genetic instability. Most of the cells in our bodies divide in order to allow for growth, maintenance, and repair of tissue. Under normal circumstances, cell division begins by the cell copying its DNA and then splitting to form two daughter cells. As the DNA is copied, genetic instability. Most of the cells in our bodies divide in order to allow for growth, maintenance, and repair of tissue. Under normal circumstances, cell division begins by the cell copying its DNA and then splitting to form two daughter cells. As the DNA is copied, there are checkpoints and multiple surveillance mechanisms in place to proofread the DNA that is being copied to make sure it's exactly like the original. Our bodies produce tumor suppressor and DNA repair genes, which regulate cell division and fix errors that occur during replication. As long as the DNA stays healthy and maintains its integrity, the cell is able to divide and perform all its necessary functions. What happens when things go wrong? Defects in cell division or in the DNA surveillance and repair processes can result in something called genomic instability. Genomic instability is an increased likelihood for mutations and alterations to occur and remain in the DNA as cells divide. Genomic instability can be due to exposure to toxic substances that induce DNA damage, or can result from diseases like cancer that disrupt the normal cell division and DNA repair processes, allowing cells to grow out of control. Two common types of genomic instability are homologous recombination deficiency, known as HRD, and microsatellite instability, or MSI. In some cancers, mutations may be present in genes important for repairing damaged DNA. When this happens, errors that occur when DNA is being copied will not be repaired. Broken, missing, rearranged, or extra sections of DNA are types of defects present in cancers with genomic instability.
What are telomeres? Telomeres are structures made from DNA sequences and proteins found at the ends of chromosomes. What are telomeres? Telomeres are structures made from DNA sequences and proteins found at the ends of chromosomes. They cap and protect the end of a chromosome like the end of a shoelace. In almost all animals, from the simplest to the most complex, telomeres are required for cell division. With each cell replication, the telomeres get shorter and shorter until they're so short that your cells can no longer divide. When cells no longer divide, tissues age. However, telomeres can be rebuilt by an enzyme called telomerase to restore cell division. If you think we've found the fountain of youth, think again. If a cell keeps dividing uncontrollably and overcomes the limitations of telomeres, a cancerous tumor can form. This is why telomeres are so important. In healthy cells, they achieve the necessary balance between limiting cellular lifespan and your own longevity and keeping cancer growth.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane separating it from the cytosol, and an inner membrane surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
In a normal cell, replication occurs until cell death. When a protein attaches to receptors on the cell membrane, it signals cellular senescence to occur. Senescence is an aging process that halts cellular replication. This causes the cells affected to stop multiplying and decrease in number. For example, if the red blood cell supply in the bloodstream decreases, anemia occurs. When cell counts decrease, age-related conditions can manifest throughout the body. Senescence may also cause neurodegenerative diseases such as Alzheimer's and dementia. Scientists are still researching a possible link.
The progression of 3D biology was driven in a lot of ways by induced pluripotent stem cell technology becoming available and widely used.
histones are not returned to the right places, and DNA methylation is added in places where it shouldn't be.
Tuins, and they control the information in the cell. In fact, mostly in the form of sugar. And then the third group is called mTOR.
South and eastward. The central and eastern parts of Kansas will very probably be cloudy in the morning, and then western Kansas will begin to get increasing cloudiness by tomorrow afternoon. Today, however, very, very pleasant reading. 73 up in the Goodland area, 72 at Garden City, 70 reported by Dodge City and Liberal, 72 over by the good folks of